Well, let's start today. One of the most interesting topic that is process in biotechnology. तो एक्चुअल प्रोसेस होती कैसा है? एक रिकॉम्बिनेंट डीएनए से रिकॉम्बिनेंट सेल बना के उसको इंडस्ट्रियल लेवल पे ऑलमोस्ट हाई प्रोडक्शन करना है, जैसे इंसुलिन होता है, जैसे हार्मोन्स, दूसरे हार्मोन्स हैं, वैक्सीन प्रोडक्ट हैं, या डिफरेंट रिकॉम्बिनेंट प्रोटीन्स है, डिफरेंट फार्मास्यूटिकल प्रोडक्ट्स है, या डिफरेंट एंजाइम्स है. How they are being produced on a huge amount, such precious and costly products for welfare of course, humankind. That is anthropogenic concept of biotechnology. Let us start कि ये बनाते कैसे हैं. ठीक है, तो सबसे पहले हमको एक जीन ऑफ इंटरेस्ट कंसिस्ट करने वाला डीएनए सोर्स ऑर्गेनिज्म से आइसोलेट करना होता है. सो द फर्स्ट स्टेप इन प्रोसेस ऑफ बायोटेक्नोलॉजी इज आइसोलेशन ऑफ द डीएनए which is of our interest or which has certain genes that can produce the product of our interest. Okay, so the first step is isolation of DNA. Now we know that the DNA exists inside the nucleus. That nucleus is inside the cytoplasm. The cytoplasm is present, covered by cell membrane and cell wall, etc. So first of all we have to separate it from the cell wall. So to do that, we first isolate the DNA by breaking the cell wall. If suppose it is bacterial cell, we are searching for DNA, we will use lysozyme, because lysozyme act on the peptidoglycan wall of the bacterial cell wall. If it is plant cell, then cellulase is being used, because plant cell wall consists of cellulose along with pectin, so cellulose is the chief material there. If it is fungal cell, then definitely we'll use the enzyme chitinase there, because chitinase act on the chitin, which is the chief component of the fungal cell wall. Let's move inside the cell. The other macromolecules we have to remove, that is RNAs and proteins. So ribozyme or ribonuclease is being used to remove or digest the RNA present from that cell, and for protein we use protease. So simply आइसोलेशन ऑफ डीएनए के अंदर हम क्या कर रहे हैं, कुछ एंजाइम स्पेसिफिक यूज कर रहे हैं सेल के डिफरेंट मॉलिक्यूल्स को ब्रेक करने के लिए. अगर बैक्टीरियल सेल है तो लाइसोजाइम, प्लांट है तो सेलुलोज, फंगल है तो चिटिनेज, आरएनए के लिए राइबोन्यूक्लियस और प्रोटीन के लिए प्रोटीएज. उसके बाद हम क्या करेंगे, जो लास्ट वीडियो में देखा था, रेस्ट्रिक्शन एंजाइम या रेस्ट्रिक्शन एंडोन्यूक्लियस, जैसे इको R1, बाम H1 या SAL2 या PUB2 या PST1, ऐसे रेस्ट्रिक्शन एंजाइम यूज करके हम उस डीएनए को कट करेंगे. तो कट करते वक्त एक हमको लॉजिक क्या याद रखना है, दैट द बोथ वेक्टर एज वेल एज द source DNA is being cut by the same restriction enzyme. Why? Because they will create the same kind of blunt end or cohesive end that will help in ligation to create the recombinant or chimera. Okay, once we cut the DNA, then that DNA is in very small amount, so we need more amount of such recombinant such source DNA. For that polymerization of that source DNA, we'll go with the procedure PCR, which is our next segment. And then we transfer that gene of interest into the vector. That vector becomes the recombinant DNA. And then what we'll do, then that recombinant DNA, our vector, we transfer it to the host cell. The host cell is one which is ideal to that vector, where that vector along with its all characteristics, that is its origin of replication, marker gene, etc., comes inside the host cell, and it is compatible to grow or adjust and multiply and replicate inside the host cell. Suppose E. coli, we use as an ideal host. Then selection of this, that selection of blue white recombinant we have seen कि recombinant हुआ है कि नहीं देखने के लिए हम X compound, chromogenic compound लेंगे, and then that recombinant colonies will turn white color on this petri plate medium. अगर non recombinant है तो वो chromogenic product will get degraded, what you call digested, and the colonies appear blue color, which are non recombinant. And once we create that, we isolate and we go for large scale production on the industrial level. Let us see simply how this process works. So we got this, the DNA. This DNA is started, it is because it is the source DNA which we have taken from the respective organism. और इस DNA में कुछ ऐसे genes हैं, these are of our interest, जो कि हमारा product produce करते हैं, say hormones or proteins or enzyme, etc. And this is our ideal vector which has markers, gene which has restriction site, marker site, everything is correct. So what we did, we took the vector like PBR322, we took the DNA from source organism, and what we did, with the help of same restriction endonuclease, we cut this DNA and this restriction and what you call vector, and then we ligated by the enzyme ligase. In this way we created now a recombinant DNA. Okay. Now this recombinant DNA it will be subcloned into what? A specific host. That host should be ideal for that vector, say E. coli. Then once we subcloned into that specific host, say E. coli, then we select the recombinant by that chromogenic XGAL method which I said just, and then inside that specific host it will increase its number, that is the copy number. ये कितने copies बनाएगा उस host cell में जितना उसका copy number है. What is copy number? Copy number is the number of that plasmid or that recombinant vector inside the cell. Say some of the plasmid has one copy number, some have two, or some of the plasmids of Pseudomonas bacteria have 100 to 200 copy number. So that number of copies of this recombinant DNA is being produced into that host cell. Then that host cell go in reproduction phase, that is the cell division, binary fission. We get these two what you call cells. They continues to multiply. How? We do the culturing of them. That is the second step in genetic engineering and this culturing in biotechnology. Culturing is being done to grow on ideal nutrient medium, and then we'll get huge number of colonies of these recombinant bacteria. फिर इस recombinant bacteria को maintain करेंगे, that is culture maintenance at proper temperature in incubation chamber. उसके बाद उसको लेंगे production के लिए inside the bioreactor, and the huge amount, lakhs of liters of the product is being produced in this fermenter or bioreactor on an industrial scale. In this way, we do the process in biotechnology. We take the source DNA, the vector, same restriction in the nucleus, cut it, paste it, join it or ligate it by ligation, then subclone into specific host, then select the recombinant and then increase the copy number, then its cell division, multiplication, culture maintenance, inoculum production and the production medium in the fermenter. In this way, the product like insulin or vaccine or proteins are being produced on a large scale in a fermenter. Now, during the discussion I said about PCR, when we take the foreign DNA of interest, it is in very small amount and we have to increase its number, that is polymerize its number. How we'll do it? That is being done by a brilliant technique developed by Kary-Mullis in the year 1983, that is PCR, polymerase chain reaction, and for this marvelous work he got Nobel Prize in the year 1993. Okay, now what is PCR? Basically it is an automated thermal cycler. मतलब इसमें heating cycle automatic continuous होते रहते हैं. Since they repeat in the same sequence, it is called cycle. And what is cycle? The heating cycle. That is thermal cycler, which is automated. It works on electric and automatic. Okay, like electric device. अब PCR करने के लिए सबसे पहले हमें याद रखना है कि what is the requirement, what it takes to increase the one DNA we got into billions of copies. Okay, so requirement is DNA template. This source DNA which we are going to polymerize into 100 to billions of copies is our template DNA. तो template DNA कैसे होता है? ऐसे होता है. DNA is double stranded, upper strand 5' to 3', lower strand 3' to 5'. In this way we got the template DNA. Then what we need? DNA polymerase. Of course हमको अगर DNA replicate करके नई copies बनाना है तो वहाँ पे enzyme acting is DNA polymerase. So we got the DNA polymerase. Okay, what we need? That DNA polymerase should be of heat resistant category, because generally most of the enzymes get inactivated at high temperature, about 30 to 37 or 40 degrees Celsius, they stop working. But there are certain heat resistant organisms like Thermus aquaticus. They can grow at high temperature. They are thermophilic, heat loving bacteria, so they can grow at 70, 90 or 100 degrees Celsius. So from such thermophilic bacteria, this DNA polymerase, that is Taq polymerase. Why it is called Taq polymerase? Because it is being isolated from T Thermus, AQ aquaticus, and P for polymerase, that is Thermus aquaticus polymerase. Then what we need? DNA primer. You should know that DNA replication or duplication cannot start if the DNA primer or template primer is not there. What is primer? Primer is just an oligonucleotide sequence of 6 to 10 nucleotide base pairs. Okay, so DNA primer अगर नहीं है तो DNA replication will not be started by the DNA polymerase. That's why DNA polymerase is highly dependent on that primer to initiate the replication process of the DNA. Then what we need? This DNA primer, remember it is a logical fact, is always selected complementary to the 3' end of this source DNA, which we are going to replicate and produce billions of copies. And then we need DNTPs. Of course, if we are constructing a DNA by duplicating it, we need the raw material, and we know that the DNA is made up of deoxyribonucleic acid is made up of that 4 base pairs, that is adenine, guanine, thymine, cytosine. These are the deoxyribonucleotides. So we need the DNTPs. Okay. Again, what we need? Buffer solution. Since this is a very keen process, it should not change its pH, and to maintain that pH without affecting the rate of reaction, buffer solution is required. Of course, for betterment of the process, magnesium divalent is मैंडेटरी देयर. अगर ये नहीं है तो प्रोसेस कंप्लीट नहीं होगी ठीक से. एंड वी नीड द रिएक्शन ट्यूब्स. दीज ट्यूब्स हैव द साइज ऑफ 10 to 200 microliter quantity, in which all these ingredient requirement are added, kept, and the automated thermocycler machine is start. तो अब इसकी प्रोसीजर कैसे होती है? The procedure is very simple. Initially, we heat the sample to make compatible or adjustment of this enzyme Thermus aquaticus, because it is a heat or a high temperature functioning enzyme. Okay, so we initiated, that is being done at 94 to 96 or 98 degrees Celsius. Then denaturation. Denaturation means simply separation of the two different strands of the DNA template. Now this becomes separated here. The hydrogen bond, that is double bond between adenine thymine, and triple bond between thymine and cytosine, guanine and cytosine is being broken, and the two template strands get separated. Okay, we have this template and we have this template strands, and this was a DNA double stranded. We got separated these two strands by the process of denaturation. It is done at 94 to 98 degrees Celsius for 30 seconds. Okay, now what will happen after this? Annealing is proceeded. What is annealing? When this primer, we know that this primer is must for DNA polymerase to start the polymerization or copying of the template strand. तो यहाँ पे annealing में this primer will come and join to the which end? Remember 3' end. So here the 3' end, here the 3' end. These two primers came. After that, what will happen? Extension, that is elongation. So continuously after this primer annealing, this DNA polymerase add the DNTPs continuously, and this strand starts elongating, and finally this becomes one DNA template, double stranded becomes two DNA, same DNA template. This cycle repeats, and within some time almost billions of copies of the source DNA can be produced. So how simple is the procedure? See, for PCR what we are doing, heating the requirement to make what to adjust, Thermus aquaticus to adjust at 94 to 98 degrees Celsius. Then we are denaturing. Denaturation is done at 95-98 degrees Celsius. Up to what time? 30 seconds. The two DNA template strand will separate by how? By breaking hydrogen bond between adenine, thymine and guanine, cytosine. Two separated strands become separate. Then what is being added in annealing? Next step, the primer comes there, at which end the 3' end of the both strand. Then extension or elongation goes on at 72 degrees Celsius. In this way, the same cycle of heating, that is thermal cycles, get continuous, automated. After each that duration of 30 seconds, 1 minute, 30 seconds, 1 minute, it continues, and in this way, within few hours, we get billions of copies of the source DNA. Because of this importance and this impact of this gene cloning method application in PCR, he was rewarded Kary-Mullis Nobel Prize in year 1993. So what are possible applications of PCR? सबसे पहले application, it can be used for multiplication or polymerization of the source DNA. It can be used for identification of unknown DNA. Suppose any pathogen, pandemic, coronavirus, okay, its nucleic acid can be detected by PCR by increasing the number of its genetic material. Again, it can be used for detection of parent and sibling relationship, because DNA matching should be there. It can be used for DNA fingerprinting. It can be used for DNA cloning. In this way, this is a marvelous process application for the recombinant DNA technology and biotechnological application. I hope you understood this. What to be remember, recombinant कैसे बनाते हैं? Recombinant cell को culture maintain करके fermenter में कैसे produce करते हैं? That is the process. और जो source DNA है उसके billions of copies करने के लिए एक automated thermal cycle use करते हैं, Kary-Mullis का बनाया हुआ, and that has particular requirements लॉजिकली क्या याद रखना है रिक्वायरमेंट में, डीएनए पॉलीमरेज स्टार्क पॉलीमरेज है, वो थर्मस एक्वाटिकस से लिया गया है. इट इज अ थर्मोफिलिक ऑर्गेनिज्म. इट रिक्वायर्स cations, that is magnesium divalent, that is must for the process. It required to complete the process. And finally the steps that alternate thermal cycle step: heating, then denaturation, annealing, extension, that is elongation. In this way the cycle repeat. So it's just a simple topic to understand. I wish you understood it correctly. Thank you. Thanks a lot.